last time we sort of finished up the story of DNA methylation. And what I'd like to do today is tell you a little bit about what we know about DNA methylation and cancer. So this is not really basic research, but you'll see that there's basic aspects to this that are probably very important for our understanding of this process. Okay, so a long time ago, maybe 20 years ago, it was discovered that there are regions of DNA that undergo de novo methylation in a number of different tumors, a number of different types of cancer. And of course, as you would imagine, the regions that undergo de novo methylation have to be CpG islands, because it's the CpG islands that are unmethylated to begin with. So if something undergoes de novo methylation, it has to be a CpG island. And so again, just to put it into perspective, these things are left open, or left unmethylated from early time in development. They remain unmethylated throughout development. It doesn't mean that they're all turned on, all these genes that are unmethylated. Some of them are repressed by other mechanisms. But they remain unmethylated throughout development. And in tumors, some of these CpG islands now get methylated. Okay? And that's abnormal because they're not methylated anywhere in the body. So that makes it an abnormal methylation. It seems to be associated with cancer. So at the beginning, before there were global methodologies for looking at DNA methylation, people were interested in tumor suppressor genes. And the idea was that if something gets, well, the idea is that in tumors you find that tumor suppressor genes are inactivated, usually by mutation. So if you take a look in every colon tumor in man, for instance, there's a gene called APC. Every single, or almost every single colon tumor that you see has APC inactivated or mutated in the tumor. Okay, so this is a typical tumor suppressor genes, and there are many tumor suppressor genes of this nature. So when they started looking at these genes, it was discovered that sometimes you find that only one allele has undergone mutation or some sort of change. So the question is, what about the other allele? And so they started looking at methylation, and sure enough, you find that many times these tumor suppressor genes are methylated in the second allele. So this sort of brought up the idea that methylation plays a role in cancer by inactivating tumor suppressor genes. How does it work? The idea, the dogma was, or the concept was, that it works similar to a mutation. That mutations work in the following manner. Let's say you're exposed to something that causes mutation, or you have something in your genome that makes repair not so good. And you get a mutation. Now the mutations are basically random. They can happen anywhere in the genome. Maybe not completely random, but they can happen in a lot of places in the genome. Right? But because there's selection, because cancer cells grow, there's selection, you end up seeing the mutations basically in the tumor suppressor gene. Right? And they said, well, maybe this is the way methylation works also. In other words, the same concept. You have something happens in cancer, in a tumor cell. There's a leakage of de novo methylation. Remember, usually you don't get de novo methylation in somatic cells. Right? They're not undergoing active de novo methylation. Right? So maybe there's some sort of leakage. There's excess de